But anyhow, I chose a topic which can be taken into different disciplines. Human microbiota. In simple, are they essential for human survival? If all participants are microbiology, probably this topic I wouldn't have taken, because they all know that they are essential for survival. But since other disciplines are there, and I also discussed with many other common people, whenever I discussed with them, they said, do you know anything about microorganism? They always used to say, oh, they are pathogens. Yes, of course, I got many diseases due to microorganism. Oh, they are bad organisms. Oh, they are very notorious organism. They made whole world to shut for 2 years. And this is how the comment I normally receive whenever I do interact with either children, either kids, either many other persons. Well, that is not the exact behavior, that is not the exact characteristics, that is not true with microorganisms. So if there are no microorganisms, I would like to say that probably we wouldn't have seen any life at all. This is one line statement I can give as a microbiologist. And of course, I can justify the same also in my future slides. And today, to justify, I am taking humans as an example by taking human microbiota. Because whenever any bacteria enters in our body, we always feel that we are sick, we are infected, we are supposed to take antibiotics, kill them, control them, take measures, clean your hand so that microorganism comes. Okay, wash your face so that you can get rid of microorganisms. Take safety measures, make sure that no microorganism enters around you. This is what which is generally present. So that is the reason why I chosen this topic, human microbiota. Are they essential for human survival? Well, when I say, yeah, this is about me. Already Shakuntala madam has clearly told about me, but still I am giving my email IDs and my contact number. If somebody is really interested in contacting me after the discussion or after my class, anybody who is interested to discuss more about this, or anybody to know something other than your aspects in microbiology, I am open for it. You can feel free to contact me either through email or to my mobile number. I am available for that. Well, whenever we were in high school days or during early education days, we were being taught that our body, a human body, is made up of with cells. Even if you ask somebody today what is the basic units of human body, immediately they say cells. And it is very clear that our body is made up of with cells. And cells is made to different organs, tissues, and many other aspects, what all we have. And this is what even I am being taught during my early education days. But recently, maybe around 2 decades back, around during the 2000, let me say very clearly, or early 2005, between that, the major scientific platforms like Nature or Scientific American or the Annals of Science or the PNAS, and of course, including the most widely circulated one of the economists, has made some headings in their cover page. This has created a major buzz around the world, especially the scientific community has really discussed a lot about these headings. The first heading I would like to take as a first step for my discussion is from the economist. It is out of the science actually, a non-science background which discusses other things, made a statement that microbes makes man. And somewhere in the same period, the Scientific American has said that your inner ecosystem, that means your body in which the bacteria are present, they outnumber your own cells. Means there are bacteria more than your number of cells. And they also asked who is in control, whether your body is in control of cells or whether your body is in control of bacteria. And at the same time, the Annals of Science has made one more heading that germs are us. Look at this statement. It's a contradictory statement according to me. We say germs which are there to kill us actually, to cause disease, to create problem. But they said germs are us. We are germs. And look at the picture which is there at the center. A toddler is being attracted by crores of microorganisms. They are moving towards a baby. A lot discussion has been taken place this. Many like us have asked, is it true? How can you make a statement like that? How can you say that lakhs of microorganisms attracted towards a baby, even though we make every day clean, we maintain sterility, we maintain cleanliness, soap, shampoo, powder, everything we apply, fresh clothes and everything. Even then, how can those all germs can come and attack the toddler? And then in Nature, a very interesting article has published that body microbes, especially they have taken a human microbiome. That's what my topic is today, human microbiota. Body microbes outnumber our own cells. And they also mentioned in Nature that if there are 10 bacteria versus one cell, it is like 1:10. If one cell is there in our body, 10 bacteria is there. It is a time we get to know our fellow travelers. This is the statement made by the Nature heading. We are supposed to understand something which are present in our body who are also coming with us. So with this small preamble, by taking into a consideration of major scientific platforms, I am taking into my lecture. Well, the concept of microorganism has started, or the subject of microbiology has initiated. We always call him the father of microbiology, a person who has laid a foundation stone for a subject, Antony van Leeuwenhoek. Due to his initial studies, today we have a subject called microbiology. But this person, during 1860 or 1800, somewhere during that time, he has seen a tiny organism. Then onwards, he has sketched them. He has made a report and communicated to Royal Society of London that I have observed something like this. But actually, he was not a scientist. He was a cloth merchant. He was a designer of clothes. He used to design the clothes under the lenses. So micro design was his passion. In order to do micro design, he used to use the lenses. And this work made him to study even tiny particles. And he has observed tiny particles which can move, which can present in different habitats. Then, with a curiosity, he started observing different samples for the presence of these tiny organisms, for the presence of these microorganisms. And in his report, he has clearly made that he observed these small microorganisms on human body. He observed like skin peelings and different parts, and also he has taken the tooth crevices, kept it under the lenses, and observed the microorganism, and he reported that he has observed microorganisms. And that's how the microbiology subject has came into an existence. Everybody was so happy. Oh, we have tiny organisms. They are present. But at that time, one of the major missing link was they observed microorganism, but from where they have observed, where were they present? This truth was completely being neglected. They somehow did not even think that why are they present in human body? Why are they present on different parts of the human body? But after almost 100 years or maybe plus, people started thinking by revamping, by looking back, the Antony van Leeuwenhoek, yes, he has observed microorganism, but on human body. So this has triggered the scientific minds, and they started looking into many parts of human body for the presence of microorganisms. Many have decided to study what kind of organisms are present, what are they doing, how are they helping, and all these aspects are started. Dear fellow colleagues, today knowing human microbiome is one of the top priority. Many research laboratories around the world, they are working on human microbiome. There is a huge progress on this human microbiome. So today I'll be highlighting the major progress of human microbiome. Well, initially, to compare the number of microbes which are present in our body, they started estimating. A rough calculation resulted to show that in our body, if we have 10 trillion human cells, it is known factor. We have around 10 trillion human cells, different types of cells. But the number of microorganisms or number of bacteria we have in our body is 100 trillion bacterial cells. It is almost 10 times more than that. Dear colleagues, if you think in a basic logic, we have 10 trillion human cells, 100 trillion bacterial cells in our body, then what is happening inside our body? Who is exactly making all these aspects? And of course, whenever we think of human cells, we also think of genes, because we all know that all our actions, activities, functions, everything are controlled by our own genes, which are present inside our body, inside every cell. So by taking into a consideration of that, we have a rough estimation that 22,000 human genes, and they also say very recently that it went up to 36,000 or 30,000 number of genes which are present in humans. But how many genes are present in bacteria? Because they are present 10 times more in our body. As these bacterial genes are also functioning like our normal genes. But we have around 20 million microbial genes. But when you compare with the number of human genes versus microbial genes, it is almost 100 times more. If somebody claims that 22,000 human genes are controlling our behavior, controlling our activities, making life functions, inside our body, there is 20 million microbial genes. They are expressing, they are working, they are also behaving like normal genes. Then what exactly which is happening inside the body? This is what the discussion has started. Well, some people started asking if microorganisms present in our body, where and all they are present? Is there any specific reason? Is there any specific place? Is there any specific organ where only microorganism present? I would say throughout our body, microorganisms are present. If we take into a consideration of major organs like nose, mouth, lung, stomach, colon, sexual organs, skin, or any other things, there are microorganisms. There is a microbiome. But wherever they are present, they are helping us. There are so many microorganisms found on nose. They are very particularly involved in mucus production. This is one of the first line of defensive mechanism. They also need to secrete antimicrobial chemicals. The microbes which are present in the nose also produces antimicrobial element. They don't want pathogen to enter. They are protecting us. In our mouth, especially we use it for the primary entry of food. The microbes which are present in our body, mouth, helps to assist digestion. And of course, they also make sure that no pathogen enters here. And there are many microorganisms present in lungs. They lubricate pulmonary tissues, helps to exchange, helps for breathing. And of course, stomach, which prevents gastritic complications. And they helps primary digestion, which takes place in stomach. And there comes one more reason in our body, colon. A major reason where high number of bacteria are present, that is the colon. I'll come back to this. Colon microbes, gut microbes. On sexual organs also, microorganisms are known to be present. They maintain pH, hydrogen peroxide production, so that they can kill the pathogen which enters through that. And of course, most importantly, our skin, which is always exposed. We always want to keep it clean. But even then, on skin, there are so many numerous microorganisms, especially bacteria, are found. And they act as primary line of defensive mechanism, which helps to control others. And there are reports which tells that every human being, individual, in their own fragrance. And of course, there are microorganisms which are responsible for that fragrance which comes out of the body, a scent production. So when we say something like that, people started asking Do you think if there is no microorganism, we don't live? Come on, sir, what are you telling? If there is no microbes, that means we can't live? This is the question is being asked. To answer this question, scientists have conducted experiments by having a concept like notobiosis. Probably you might be knowing that. It is a concept, notobiosis, where they create a sterile area or sterile organism. Sterile refers to germ-free, aseptic, axenic, sterile, no intervention of microorganisms at all from the beginning. They have a model organisms developed in that model in that condition, and those model organisms are said to be notobiotic animals. A fish has been developed, it is said to be notobiotic fish. A plant is being developed in a sterile condition, notobiotic plants. They made sure that from the beginning till the last, there is no association of microorganisms. They made it clear that no bacteria, no microorganism should come in contact with these model organisms. But the results of the same, which clearly tells that all these notobiotic animals or fish or plants, they have shown a very poor survival rate with lot of complexities. They have shown abnormal functions. They have shown abnormal behavior. This is very clearly states that without microorganism, we cannot expect a normal life. Well, about the gut microbiota, where, as I said in my previous slide, that there is a gut microbiota, there is a colon. Until 1990, this gut microbiota concept was not very much famous. And in this reason, major amount of microorganisms have been found, including bacteria. In the gut alone, more than 1,000 species of bacteria and other organisms have been derived, not just one species. More than 1,000 species. Over 7,000 strains have been recovered from the gut region. And this has led to think twice why these organisms are present in gut. What are they doing? That's how gut microbiota concept has initiated. And in the gut, there is more than 80% bacteria or microorganism present in that gut. We are unable to culture it actually. We don't know what do they eat. We don't know how do they behave and all these aspects. So that's where the prominence has been given to understand more about gut microbiota or human microbiome. If I take into consideration that the microbial diversity which is present in gastrointestinal tract are the total reason, like we all know that there is a stomach, there is a small intestine and a large intestine. Large intestine is typically called as colon. From stomach till the rectum, the end of that, scientists have observed high number of microorganisms, including bacteria, archaebacteria, eubacteria, like different types of bacteria, and different types of microbes, including fungi. And among that, the bacteria have found more than 90%, means dominant ones are bacteria rather than other microorganisms. When they have studied the habitats of these colon to know the number of microbes which are present in that region, they found that in stomach alone, where probably you all know that the pH is somewhere near to 1 to 2 actually. Sometimes it can go up to 4, like it depends on the intake. But highly acidic in nature. But in that condition only, the number is 10 to the power of 4, more than 10 to the power of 4 per gram of the region. Then if we take into a consideration of small intestine, including duodenum, jejunum, and ileum, the number of bacteria which are present immediately goes up. Like initially, it says 10 to the power of 2 to 10 to the power of 4. Then when it reaches to the ileum, where the pH increases from acidic to neutral, there you see 10 to the power of 7 to 10 to the power of 9. It's a huge increase in number of bacteria which are present per gram. And next comes, which is popularly called as colon. In microbiological terms, we say it's a bioreactor, a fermentative device where anaerobic fermentation takes place. But suddenly, in that colon, in the region where a fermentative characteristics are present, the number increases from 10 to the power of 4, 10 to the power of 7, but here it goes 10 to the power of 11 to 10 to the power of 12. And guys, this is the reason where the major metabolic activity takes place. This is the place where most of the undigested food gets digested. This is the place where microbes are highly active. This is the place where the short chain fatty acids are being developed, which will be essential for our body. And of course, even in other parts, in other pathways are also being produced, but this is the place where major short chain fatty acids are being produced. But when we take into a consideration of all these, if you can look the different types of bacteria which are present, which is given at the bottom. In the stomach, there is a lactobacillus. In the small intestine, there is a presence of lactobacillus. And of course, in colon, there is a lactobacillus. One of the common organism which is known to be present, along with others. But I am taking lactobacillus here. You will let you know. You will get to know why I took lactobacillus here as an example. Well, when they are present in the gut, they are known to perform many functions. They help in breakdown of foods, like both digestible and undigestible. They supply the gut with energy, like metabolites and secondary metabolites. Both are being produced by the microorganisms. And here, generally, the gut epithelial cells derive energy from microorganisms. And probably you might be knowing that we do have the ability to synthesize many vitamins. And gut microbiota are also known to produce or make vitamins like vitamin K, B12, folic acid. And these organisms also train the immune system, like which organisms are pathogen, which organisms we are supposed to control, which organisms we are supposed to fight against. So this mainly helps in the immune system to differentiate between pathogens and the beneficial bacteria. And of course, these organisms are also known to break down of toxins, especially the endotoxins and mycotoxins. I am stressing the word mycotoxins here because my research lies in this point. The gut microbiota are a part of gut microbiota are the kind of organisms which are present in gut microbiota also known to degrade mycotoxins. Now, this is how my research is being connected with gut microbiota and mycotoxins. And of course, along with that, they protect against pathogens because they compete with pathogens whenever it enters by colonizing the epithelial layer, the outer layer, and also by secreting many antimicrobial compounds. And recently, there are so many results or research which are telling that there is a gut-brain axis. There is an interaction between gut and brain. Anyhow, I don't want to speak much about that because many research are being currently taken out to know the interaction between gut microbiota and brain. So when organisms are present in our body, something like this, what if we lose these organisms? There is a high probability. They are present inside body unknowingly, whether you want it or not. They have entered in our body. They are working for us. But some of the activities performed by us are also making losing of this organism. That's what is called dysbiosis. Change in population and diversity of microorganisms present in our gut or in our body. Well, dysbiosis is accepted if they are pathogens. We want to kill them. Like many times, we all have taken antibiotics to kill bacteria which are present in our body. We want to kill. But if they are pathogens, we can get rid of these, but they are not pathogens. More than 85% of good bacteria present are good bacteria. So dysbiosis is happening. We are losing these good bacteria. Due to a dietary change, suddenly we started following Western culture. We started eating pizza, burger, roadside foods, unwanted things, which is not being followed right from the beginning. Our food culture is getting changed. Drinking more alcoholic beverages, taking unprescribed over-counter antibiotics. Many times I have seen people who take simply antibiotics. If they get throat infection, immediately they go and take azithromycin, azithrol 500, or any other antibiotic. They don't even think whether this is due to bacteria or something else, whether due to allergy or something else, or due to viruses or something else. Simply they take antibiotic. And of course, the modern age activities like high level of stress and anxiety makes a huge metabolic alterations. That is also leading to dysbiosis. Then what are we supposed to do to maintain these organisms? restoring the balance of gut microbiota. This is where the major aspect comes about my research. We want to protect them. We want to balance them. We want to maintain them in our body. Then what are the ways in which we can maintain? Is mainly by balancing our food. Today, I think probably you might have heard many talks. You might have read many lines, headlines, where everybody started telling, take good food. One of the major thing which is causing so many problems. So balancing of food plays a vital role in maintaining the gut microbiota. And probiotics. If you can remember and recall that in the stomach, in the small intestine, in the large intestine, there are bacteria like Lactobacillus. Most of the lactic acid bacteria are known to be act as probiotics. We can take probiotics to restore our gut. And very recently, to avoid dysbiosis, the FDA, Food and Drug Administration USA, has approved fecal matter transplantation, FMT. I don't know how many of you have heard, or this was a major headline actually. I have seen in local newspapers also in India, though it is approved in USA, but it is already available in the market. Fecal matter transplantation. Is it something like eating our own shit? Not exactly like that. It is not like that. People have, whenever there is a dysbiosis, whenever we started losing our own bacteria, which normally goes in the fecal matter, they have collected all the bacteria, isolated, purified, standardized, and kept it in a pill, like a tablet, and they asked people to take that only. So that's what fecal matter transplantation. And this was approved somewhere in November 2022. But anyhow, I'll not.
322 runs daily.
Jaida. Okay. So I'm not discussing more about this fecal matter transplantation, neither balanced food, because my research mainly lies on probiotics. Well, for another 45 minutes, I'll be discussing about that. So what are probiotics? They are bacteria. They are not just bacteria, they are useful bacteria. They're found in food, the fermented foods, what we eat. They can live in human gut. That's what we have discussed already. The WHO has given a definition for probiotic organism that live beneficial microbes which when administered in an adequate amount exerts health benefits to the host. Means it is a live bacteria. Certain adequate amount has to be given, then only it can exert the beneficial effects. The number has to be maintained. By looking into this parameter, these organisms have been given GRAS status, generally regarded as safe status. Most of the time, if a tag comes as probiotics, we need not to think. It is accepted. We can eat, we can take. No need to think of safety actually. At that time, when it is being said as probiotic, can we call all microorganisms or all lactic acid bacteria as probiotics? And of course, no. whenever any bacteria which is being isolated from fermented food, which is being isolated from different habitats, we are supposed to characterize them for set of attributes to say they are probiotics. Like the isolated bacteria, whether it is useful or not, first of all, we are supposed to see harmless for ingestion. The given bacteria is supposed to go and colonize, adhere to our mucosal membrane, which is present inside the stomach. The moment when it enters into the stomach, it is supposed to resistant to acid and bile, which is known to be present in the stomach. Most of the bacteria which are unable to tolerate acidic condition and of course bile salts, it dies. And then we are supposed to check for colonization, like different attributes have come. The people have studied these bacteria, looked into their several different applications. Nowadays, probiotic bacteria is used for many purposes, like to prevent diarrhea, to treat allergy, to treat lactose intolerance, as immunomodulators, for many therapeutic applications. So this is how I have attracted towards the probiotic bacteria. I've collected so many different fermented foods which are known to be uncharacterized, means for probiotic purpose, but they are well-known food, like Neera. Probably you might be knowing that. A coconut sap, water. Different fermented foods like idli, dosa, vellapam, posa, pola, curd sample, and different fermented juices also we have collected. We have tried to isolate probiotic bacteria which are known to be present. So we could able to isolate a high number of bacteria which are known to be present in these samples. In order to isolate, we use a selective media. Means selectively we wanted to isolate only lactic acid bacteria by presuming probiotics. And whenever we isolate good number of bacteria, we maintain in our lab. As on today in our lab, we have a library of uncharacterized isolates, isolated from food sources. We have around 251. We are maintaining that library, both bacteria and as well as yeast. And whenever we are interested in looking or characterizing these organisms, so we design an experiment by taking one problem. Say, one PhD student or is very much interested in knowing a probiotic bacteria which can mitigate the mycotoxins, which can control the mycotoxinic fungi. Because I am a guy who worked on mycotoxinic fungi. I know the fact that there is a mycotoxin presence in the food. There is a problem of mycotoxin which are leading to many complications in human body. Then I started working use of these probiotic bacteria to mitigate these mycotoxinic fungi and of course other pathogens, human pathogens. And this is how I started my research. So initially, whenever I decide, my students get access to the library, they start screening these library against our target research. Before even preliminary characterization, we wanted to know whether this organism has activity or not. Then we do preliminary characterization. Then we do probiotic characterization, whether all attributes present or not. Then we take it into a further aspect. So preliminary characterization, preliminary screening plays a crucial role for my students to take their work for further aspects. Like today, I'll be just highlighting in our lab what exactly is happening, maybe for around 10 to 15 minutes. So, well, we started screening and we got one efficient isolate which can control a mycotoxinic fungi. Then we do probiotic attributes like tolerance to acidic pH, bile salts, antibiotic susceptibility, or hemolytic activity, the safety precautions, the cell surface hydrophobicity. It can resist to that and it can colonize there. And then the different antifungal activities also as an additional attribute. Along with probiotic organism, it should have antifungal activities. So we do set of experiments, like we try to grow together and look for what's happened, whether the growth is controlled, how much fungal growth is being controlled. Can we control the fungal growth in different medium? Can we target the fungal progenies like Conidia? They are the progenies. They are the daughter cells produced by the fungi. So if we can able to kill them so that it is not going to spread. Like it should have the mycotoxin biodegradation ability too. We also look their ability to degrade or transform or make it inactive, the mycotoxins. Well, this slide tells one of the paper which is being published in Food Bioscience in the platform of Elsevier, wherein we have isolated, characterized one probiotic organism, that is Lactobacillus plantarum, from our own laboratory, and which is being named, and studied against a mycotoxinic Aspergillus species, especially the Acacius. As I said initially, that whenever we are interested in knowing the preliminary activity, that this is what we do. We try to know whether this can control the fungi or not at the preliminary level by doing different methods. Either co-inoculation, either line of streaking, either agar overlay, otherwise well diffusion, otherwise spot inoculation. I think probably you can see one or the two figures here. We try to confirm all the ways. And once we confirm that this bacteria can control the growth of this organism at different levels, at different targets, then we look for their ability to inhibit the germination of Conidia. Whenever the fungi has produced, whenever the fungi has grown, immediately it is known to produce spores actually. And this fungi, this bacteria, Lactobacillus plantarum, should also have the ability to stop or to kill the Conidia. That's what Conidia germination inhibition assay. So here at the bottom, you can see very clearly showing that if you compare the control, wherein control has shown the germination of Conidia, but the treated Conidia has stopped the germination of Conidia, both bacteria as well as the metabolites which are being produced by this bacteria, Lactobacillus plantarum, which is known to secrete several different metabolites. And they are known to produce bacteriocins, antimicrobial peptides, antimicrobial substances, And we were also interested in knowing whether it can produce any metabolites. And of course, it has the cell-free supernatant of bacteria has shown to produce different metabolites. And that is also being treated against conidia and of course mycelia. As I said, when we treat bacteria versus fungi, is it really exerting the action? That's what we were very much interested. When we subjected a fungi treated with a probiotic organism, observed under SEM, the scanning electron microscope, and we observed different structures of a fungi. Like when we see a fungi, there is a mycelia, there is a reproductive structures, there is a conidiophore, there is a vesicle. At what structures our target bacteria is acting upon? And we observed that the mycelia was surrounded by bacteria. There is an interaction between accumulation of bacterial cells around the mycelia. The reproductive structures, conidiophore is deformed. There is a damage in that. And of course, in vesicles, and low number of spore production. And of course, whenever there is a damage, whenever there is a deformation, whenever there is a natural no growth, and automatically results in less number of conidia production. That's what we could observe. And later onwards, we have used these bacteria and its metabolites, whether it can exert the same activity in different models. Like the Aspergillus problem is more in poultry feeds. The contamination of Aspergillus problem is there more in peanuts. So what we did was, we have taken the poultry feed and of course the peanuts. Artificially, we have inoculated. Challenge inoculation was done. We have inoculated the spores of Aspergillus into the poultry feed. And in another set of poultry feed, we have just given the metabolites or the number of bacteria along with the fungi, whether it can grow, whether it can die, whether it can control, whether it can decrease the number of fungal cells. And somehow the results very clearly show that there is decrease, there is a control of fungal growth. And of course, the same is being tested for mycotoxin production, which is known to produce ochratoxin. In control, we could see a large or high production of mycotoxin, ochratoxin, but the treated ones have shown the reduction in that. And in some, no mycotoxin production at all. We could show that. This is one of the paper which is being published. Parallelly, like again, I don't want to repeat the concept and the flow of work and the methodology what we have followed. This time we have taken one more lactic acid bacteria, that is Lactobacillus brevis, against one more mycotoxinic fungi, Fusarium verticillidis, which is known to produce one more different mycotoxin, that is fumonisin, wherein high fumonisin occurrence, and of course, there is a major problem of Fusarium in maize. So that is the reason why we took up this. And typically we followed the same pattern what I explained previously. And the same kind of results is being observed so that we could able to control the growth of Fusarium verticillidis and also the mycotoxin produced by the Fusarium verticillarius, that is fumonisin. There is a low level of synthesis, and there is a direct degradation. So likewise, we have started our research work. Currently, we are working on the same line. We have also started one more Aspergillus species, which is known to produce aflatoxin. Then, along with fungi, we started thinking about bacteria, the pathogens, which are waterborne, foodborne pathogens, need to be controlled. There is a huge problem with these organisms, Salmonella, Enterococcus, and other organisms. We started screening our library against different bacterial pathogens. But the design of experiment and the concept what we have achieved in antifungal strategies is completely different here in antibacterial strategies. We were supposed to design different experiments than fungi. And of course, we have achieved that. And somehow we have ended up in publishing this paper also by mentioning the Lactobacillus plantarum, which is a different isolate, has shown in multiple ways it can control two different pathogens, Enterococcus faecium and Salmonella. We have tried to look the activity of these organisms. Well, so we got some isolates which are having antifungal, which are having antibacterial, and we were very much interested in knowing these probiotic bacteria. They are showing antifungal attributes. They're working against different mycotoxinic fungi. They also mitigate different mycotoxins. Do they contain any extra genes, additional genes which are responsible for the antifungal activity? Because not all probiotic organisms have exerted the antifungal attributes. They were not efficient enough when we have screened our library. The potential isolates were subjected for whole genome sequencing. We have sequenced two different bacteria in order to know the different genes present in that. So this is the data for one bacteria, Lactobacillus brevis, which is currently deposited at NCBI GenBank. The whole genome sequence is available. We could able to publish this also. The data on whole genome sequencing has resulted to know many different genes. These are all the number of genes which are present in that bacteria. But we were very much interested in knowing, out of all these genes responsible for different functions, which genes are very much interested for antifungal attributes, the functional attributes of our work, which has resulted in showing that these organisms known to carry certain set of genes, especially the type 3 polyketide synthase gene, T3PKS, one of the genes which is responsible for the antifungal activity exerted by the probiotic bacteria. We are in the process of categorizing this gene at present. Our lab is targeting the role of T3PKS. And this has become, so far what all I have explained has become the platform for us to end up in T3PKS. And of course, the other different genes which are present in these bacteria, which are responsible for bacteriocin activity like lantipeptide, bacteriocins, polyketide synthase, like different genes, we got to know that we are supposed to work upon that. Well, one more student has given, we have characterized everything in the laboratory. It has shown that it can control a mycotoxinic fungi, Salmonella, in the laboratory conditions. Can we expect the same in animal models? This is the question being asked. Then we have took up these organisms, especially one which is having high antifungal attributes and one which is having activity against Salmonella, both studied at in vivo level, means using animal models. For poultry feeds, we have used poultry, like directly we have used chicken, a live chicken, taken around 120 animals, fed mycotoxin, given mycotoxin, because in poultry feed, they do add toxin binders. We subjected our bacteria, we replaced our bacteria as toxin binder. So in the same line, I got a funding from DST, SERB, in order to study lactic acid bacteria as toxin binders in poultry feed. Currently, this research is ongoing in my lab. Our preliminary results have shown that the toxin can be mitigated, controlled, or managed if we can give lactic acid bacteria or a probiotic bacteria to a poultry so that we can get rid of toxin binders, we can get rid of the problem of toxin, and of course other pathogens. The other details, anyhow, I do not have time to discuss how we have done this experiment and what we have proved in this experiment, how these poultry were fed and analyzed, what are the different parameters we looked in. Probably if Shakuntala Madam gives another chance and if I get a chance, then I'll be discussing what we did in vivo, like using animal models. But one thing I would like to mention in this result, we could show that there is a control of toxin. We could show that there is a control of Salmonella. But the question is, there is a gut microbiota which is already present in the gut, might have exerted this activity. This is the question which is being asked by the reviewers. How do you say that your bacteria has exerted these characteristics? How do you say that the bacteria which you fed has shown all these activity? For that purpose, we have isolated the bacteria from the different groups of poultry, different regions of poultry, different samples of poultry. In high number, the bacteria which we fed being isolated back in high number in different conditions. And again, we have characterized that bacteria. We have molecularly identified that bacteria. We confirmed that the bacteria which we have given has shown the activity, has exerted the activity since it is present in high number. This is what our final conclusion. So that a probiotic bacteria can be isolated from food. And if you are really eating well, the different kinds of food, it can also enter here and it can colonize and it can result in exerting better activity. So with that note, I would like to end my lecture by saying that maintain a healthy gut, lead a healthy health. I hope you could follow me, whatever I am trying to discuss and what exactly I am working in my lab. So the data which I presented being published in different publications, and all these publications as a result of my research scholars, whatever I spoke today, it is not the work what I done actually. It's joining of hands by research scholars, especially I am fortunate enough to get very good research scholars who can work. As a result of that, today I am speaking today. And of course, without funding agencies also, probably I wouldn't have done all this work. I would like to thank research scholars and of course funding agencies and my research collaborators as well. They helped me to take into the next level the genome sequencing aspect. Many different set of experiments is conducted during my official visit to different universities. So with this, I would like to thank one and all who are present there, who listened to my a small discussion wherein given me an opportunity to discuss what's happening in my lab. Thank you one and all. And of course, Shakuntala madam for identifying me and giving a chance to discuss with you all. And if at all, if any questions are there, I'm very happy to take.
Thank you, sir.
Sir, I want to ask one question. Dr.
Yes.
Jayashree from Hubli.
Yes, ma'am, please go ahead.
There is a one news in newspaper and Facebook. A virus from China is coming and it is hitting to a human life. Is it?
HMPV.
Yes, sir. Is it true or false?
Ma'am, there are many viruses are present. There are many viruses are emerging. Even in Kerala also recently you have seen many newses. There was Zika virus. In Shumaga, you have seen there is a Kasnur forest virus. They comes and go, ma'am. Don't worry. Even COVID also, if there was no media, if there is no active TVs and active information, probably it would have come and gone.
Okay.
There is a possibility of emerging viruses, presence of viruses, and coming and going, ma'am. No need to think too much about this HMPV. I just followed the news yesterday and few days back. I don't think so. Even I would say COVID-19 was one of the weakest virus actually. Virus did
Sir,
not.
why only? Why
Virus,
only bad
yeah.
viruses? Why not good viruses?
Ma'am, there are many good viruses, ma'am. That's what I said. We think of only bad viruses. We know COVID-19, but there are so many bacteriophages, there are so many virophages, there are so many good viruses are working for us. We don't think of them. We always remember our foes. Enemies are more remembered than friends, ma'am. Normal human tendency.
Yes, sir. Thank you so much, sir.
Yeah,
Thank you.
you're welcome, ma'am.
Virus only dangerous pathogen.
I would say, as on today concept, most dangerous or notorious organism, if at all, if it is there, that is virus.
Yes, sir.
The reason is, even if there is nothing, like it always requires a live host for its activity. Otherwise, it is said to be dead. So there is a bridge between living and non-living. Whereas there is a question whether it is living or non-living. When it comes inside our body, they are living. When they are present outside, they are non-living. This is the major problem, ma'am. So I would also agree with you, notorious organism in future, if at all, if we see, that is viruses only.
Wow.
There are so many facts to justify this. Hope
Sir,
sir,
actually, I have one question, sir.
Yeah, please, Govardhan, sir.
Sir, how can we isolate some probiotic bacteria from various food sources if they are obligatory for the particular source?
No.
If they are growing
Yeah.
in media means,
Yeah. So preferably it is known to be present in fermented food.
yes,
So in curd, if you take, we do add a small amount of curd or buttermilk to milk. Okay, then it makes as a curd actually.
yes,
If you are interested in isolating bacteria from curd, take a curd and prepare one media called MRSA, which is available, which is selective media to isolate probiotic bacteria. Take that MRSA media and simply serially dilute this curd sample and inoculate onto the Petri plate MRSA.
yes,
100% you will identify bacteria, probiotic bacteria from curds, because curds is one of the excellent probiotic food in the world till today.
yes, sir, yes. So, but what about other food sources like you told, Jijugo
Yeah,
and
see, whatever
Nira?
source is there,
Yes, yes,
what if it is fruit, or if it is juice, or if it is any idli, dosa, whatever is there. Okay,
yes,
preferably in idli, dosa, prepared food, you cannot expect bacteria because you are going to kill them by with steaming. When you prepare idli, most of the bacteria gets killed actually.
yes,
But you can ask then, what is the use of eating idli actually? Still, idli is good because the metabolites will be there, produced during the fermentation process.
yes,
So
sir.
if you want to isolate bacteria from idli, you need to take idli batter than not the idli actually.
Yes,
So
sir.
take idli batter and then isolate bacteria.
Yes.
I have one question.
Thank you, sir.
Please, sir.
Sir, you are saying the isolation of probiotic from butter or
Yeah.
curd. This is only for MRSA medium or any other mediums are available for isolating for some other sources.
Sir, better MRSA, sir.
MRSA for isolation of probiotic.
Yes, sir. Lactic acid bacteria. So lactic acid bacteria isolation can be done in MRSA media, which acts as a selective growth. Means it allows only that bacteria to grow, because these fermented foods are known to have many different bacteria, sir. We don't want to isolate all of them. We want to isolate only lactic acid bacteria. Then you go for MRSA.
So what about the other fermented milk they have for some other probiotics? That is possible for isolating for the same media or not?
No, from same media you cannot isolate other probiotic bacteria. Like Bacillus acts as one of the probiotic bacteria. At that time, you are supposed to go for different media.
That is the things I am asking. What are the kind of media, selective media for isolating the probiotics?
Yeah, MRSA is most preferred one. If not, you can go for nutrient agar, casein hydrolase agar. Otherwise, you can also think of subroots dox agar, SDA, for probiotic yeast isolation.
Thank you, sir.
Welcome, sir.
Sir, what do mechanism bacteria follow to accumulate fungal toxins?
What is the mechanism they follow? Yeah,
Yes.
well, this is being asked in our research only. So how exactly they mitigate the mycotoxin? So to answer for this, we started characterizing the CFS, metabolites which are produced by the organisms. So when we have tested the metabolites, whether does it known to contain organic acid or peptides or proteins or different enzymes, then we got to know that there are certain organic acids, very specific organic acids, which could interact with them and makes it detoxification.
Okay, so this organic acid react and remove the toxin part.
Yes, they interact with them, then they nullify the problem actually.
Okay, thank you.
Okay. And other, like there are other antifungal metabolites which can control the growth of fungi itself. They don't allow fungi to produce toxin at all.
Got it. Thank you, sir.
Yeah. I hope if there is no more questions, I think next speaker is waiting. It's already 3:30. Thank you, one and all,
Thank you,
for
sir.
your patience.